and welcome to our annual Kuan-Teh Jeang Memorial Lecture. You may have noticed that it's no longer Asian American, Native Hawaiian, Pacific Islander Month, but it's sort of a running joke that there are so many accomplishments by the AA-NHPI community that we can't fit them all into 1 month. So for the last 2 years, we've actually had our Jeang lecture in the first week of June. So I'd like to start off with some remembrances of Kuan-Teh Jeang, in whose honor we're having this talk. Next slide. Oh, I can do that. Never mind. Okay. Teh was born in 1958 in Taichung, Taiwan. He spent his childhood in Libya and then came to the United States in 1970. So he was, in many ways, a true citizen of the world. He attended the Massachusetts Institute of Technology and earned his MD PhD from Johns Hopkins at age 25, then went on to study gene regulation and gene expression of cytomegalovirus with Gary Hayward. He came to the NIH in 1985, actually the same year I did, and did a postdoctoral fellowship with the late Dr. George Corey. And that's actually another connection we have. George Corey is actually the reason that I ended up at NIH. He gave a wonderful talk at Hopkins when I was there and said, NIH is the most wonderful place to be if research is one of your top priorities. There's no better place than the NIH Intramural Research Program. So after his postdoctoral fellowship, he moved to the National Institute of Allergy and Infectious Diseases and became chief of the molecular virology section in the Laboratory of Molecular Microbiology, where he studied human immunodeficiency virus and a related virus, HTLV1. He was the editor-in-chief of Retrovirology, a leader in the NIH Virology Interest Group, a leader in the Society of Chinese Bioscientists in America, and a big advocate for racial minority groups and persons with disabilities. And we continue to cherish his memory and legacy. This is a picture of his family. His family couldn't be here today because they're actually in Texas celebrating the end of the residency of one of his daughters. Well, and if you get a chance, we actually have a memorial garden in between Building 31 and Building 33, and you can see that at your leisure. I'd now like to turn over the podium to David Rice from our Office of Equity, Diversity, and Inclusion, who would like to say a few words about the occasion.
Thank you, Dr. Owens. It's my pleasure to be in here. When I was reviewing your slides earlier today, I saw you and Dr. Jeang come here in 1985. That was when I was born. And my birthday is actually June 6th of 1985. So it's great to see that the work that's been done here in NIH has been here for a very, very long time and many, many years to come. But first, I would like to extend thank you again for allowing us to be here and the invaluable partners of the Office of Intramural Research. Your support and collaboration with the Office of Equity, Diversity, and Inclusion are deeply appreciated. As we wrap up Asian American, Native Hawaiian, and the Pacific Islander Heritage Month, it's important to reflect on two key points. First, we celebrate Dr. Kuan-Teh Jeang, and then celebrate his brilliant career. While he was here during my tenure here, I never had a chance to meet him. However, many of you here in the room have, and online today, have been one of the leading scientists in virology. He dedicated his life to science, but he was also a fierce advocate for representation for Asian Americans in leadership positions. At the Office of Equity, Diversity, and Inclusion, we are committed to continuing this legacy, and by developing strategies and working with our partners across NIH and the biomedical research enterprise, and create pathways for future generations for Asian Americans to achieve his vision for more Asian American in scientific leadership positions. Some of you may or may not know, back in September of 2022, our office, in partnership with the National Cancer Institute, the National Institute of Mental Health, and the National Institute of Minority Health and Health Disparities, published an article on the Frontier Journal examining the Asian American leadership gap. I encourage each of you, if you haven't seen it, go ahead and review the article. But I wanted to highlight one of the key recommendations from the article, which was to improve workplace culture to reflect Asian American workplace experiences accurately. This recommendation leads to my second point of reflection. This year's Asian American, Native Hawaiian, Pacific Islander month theme was embracing belonging, capturing the dynamic balance between honoring long-standing tradition while embracing new ones and embracing new ones. There are, as you've seen in the month of May, there were numerous of events across NIH. For instance, the Insight lecture by Dr. Vivek Murthy, where we explored the crucial importance of developing social connections. Additionally, just this past week, or I believe on Thursday, the Belonging Men event hosted by the Korean Women's and Science Association last week, which was amazing. I was there in person, seeing the music, the food, and the science discussion that was involved. There were many other events like this across institutes and centers that contribute to the success of this year's Heritage Month. These are a part of our office's commitment at NIH to improve the workplace culture for Asian American, Native Hawaiian, and Pacific Islanders. However, our work and commitment cannot stop after 1 month. We must embrace the idea that work culture should reflect the experiences of not only Asian American, Native Hawaiian, and the Pacific Islanders, but all underserved and underrepresented communities every single day of the year, 365 days and 366 days on a leap year. In conclusion, while we celebrate the accomplishments of Asian American, Native Hawaiian, and the Pacific Islander community, we at the Office of Equity, Diversity, and Inclusion want to emphasize that we plan on carrying out his legacy. We aim to ensure that future generations of Asian American, Native Hawaiian, and Pacific Islanders have an equitable pathway to scientific leadership. Our call to action for each of you here is to help NIH carry out this mission by ensuring the workplace culture for all communities is acknowledged and respected. This starts with everyone here, everyone online, and everyone within the biomedical research enterprise. Again, thank you, Dr. Owen, for allowing me in the office to participate today. I'm going to hand it back over to you to introduce Dr. Jenny Ting.
It is my pleasure to introduce our 2024 Jeang Lecturer, Dr. Jenny Ting. Dr. Ting was born in Taipei, Taiwan, and also lived in Bangkok and Manila before coming to the United States. She graduated from Illinois State University with a BS in medical technology, followed by a PhD in microbiology and immunology from Northwestern University. She did her postdoctoral training at the University of Southern California and Duke University, then joined the faculty of the University of North Carolina School of Medicine in Chapel Hill. She currently holds appointments as the William Rand Keenan Professor of Genetics, Director of the Center for Translational Immunology, and co-director of the Inflammatory Diseases Institute. Dr. Ting's laboratory has broad interests in the application of cutting-edge ideas and technology to the study of disease-relevant issues. Major directions include innate immunity, dendritic cell function, cell death, signal transduction, gene discovery, and a plethora of other areas. Clinical issues of interest include multiple sclerosis, cancer, autoimmune diseases, infection, and inflammation. Dr. Ting has over 300 scientific publications and numerous awards and recognitions, including being president of the American Association of Immunologists and election to the National Academy of Sciences. Her lecture for today is entitled "The Broad Impact of Innate Immune Receptors in Viral Infection, Cancer, and Autoimmunity." Let's give a big NIH welcome to Dr. Jenny Ting.
Thank you so much, Dr. Owens. It's such an honor to be here to give today's lecture. Let's see. I've been at NIH many, many times, I think probably over 40 times, so I got to know this institute and really love the people who are here. And today, like I said, it's a great honor to give the memorial lecture for Dr. Kuan-Teh Jeang, who is also from Taiwan and went through a circuitous route to the U.S. He lived in Libya, as Dr. Owens was saying, and his many credentials. Obviously, a genius-level researcher, got his PhD MD at the age of 25. He married his wife, a fellow graduate student, and these are his research, starting from his work on CMV gene regulation and later coming to NIH. As Roland was saying, he was at NIH for 27 years and major seminal discoveries about the role of TAT and also in HDLV and later on small regulatory RNA, which was blossoming. And I'm sure he would have made a ton of contributions in this area. Like was mentioned by David as well, that he was a champion of the API and president of the Society of Chinese Bioscientists in America, well beloved by his trainees and published extensively and has 3 children. So to celebrate his life and also to celebrate diversity in general, and I've learned a lot today from just talking to Dr. Owens about the mission that he and his organization at NIH has. So to really remember this wonderful scientist who was so productive and so giving of himself. What I would like to talk about today is really concentrate on innate immune receptors. And I might talk about a little bit of our other work, but mainly focus on innate immune receptors, something that we've worked on for over 20 years. So when we looked at the innate immune system, there are many receptors that are on the cell surface. Most famous is the toll-like receptor and C-type lectin receptor. However, what's really phenomenal in immunology, because in most other fields, such as cell biology, we think about receptors are being on the outside, facing the outside world. But it turned out for cells, just like for us right now, there's great danger on the outside, but inside there's also a lot of dangers. So the system within the cell has to really detect all of these danger signals, as well known at NIH. So what I would just like to tell you is our interest in the innate immune receptor started from here, which is a family of proteins that we did quite a bit of work on, which is nucleotide binding leucine-rich repeats, also known as synaptic receptors. I will tell you a little bit about DNA sensors, including some of the ones here, just mentioning them. And I'll tell you about the broad impact of these receptors on many diseases, such as in cancers, such as in inflammatory diseases, and infectious diseases, of course. So our venture into innate immunity was completely unexpected. We were initially really interested, when I was a postdoc, in gene regulation in the immune system. And Mary Carrington, who's here, actually, I think we were a year apart in our research experience, and she was so nice to join my group, which was nascent and no promise at that point, and looked at MHC gene regulation. And in the process, after many years, we started working on this gene called C2TA, which stands for class 2 I lost a few things while I was inserting my, called C2TA, which is responsible for transcription of class 2 MHC genes, this protein MHC, C2TA, which is a remarkable protein because it's a master regulator of all MHC, whether it's class 2, whether it's invariant chain, which is DN, et cetera. So this is a remarkable protein, and when you don't have C2TA, what happens, I don't think my, let me try this. Oops. I was trying to do the laser pointer. Laser pointer is not working. That's okay. I can.
Try this.
Okay. Thanks.
Yeah, the top one.
Okay. So C2TA over here, and mutations, loss of function mutations in C2TA leads to this type 2 bare lymphocyte syndrome, where the patients do not express class 2 MHC on the lymphocytes and macrophages and dendritic cells. As a result, these patients are very severely immunodeficient. Now, our work was mostly on transcription regulation, and then 2 people in my lab decided to see if there are other genes that look like C2TA, and they used 2 different domains, because if they used just one, they wouldn't have found this family. And using the nucleotide binding domain and leucine rich repeat of C2TA, they came upon 22 genes with different N termini that we earlier called caterpillar, because we want to be very inclusive and included all of the domains of these proteins. However, it is now known as NLRs, standing for NBDLR proteins, or for nod-like receptors. So what is remarkable about this family is actually found initially in plants. In plants, these are called disease-resistant genes, and some plants have hundreds of these genes, and they're mostly to detect changes in the environment as well as pathogenic changes. And in humans, there are 22 such genes, and they all have what's called a NOT domain, standing for nucleotide binding region as well, and a leucine rich repeat down here, and they have different N termini. So these are the 22 NLR proteins. Some have many of these proteins, such as the NLRP protein. There are at least 11 to 14 such NLRP proteins. Most recently, it was found that prokaryotes, such as E. coli, also have these proteins, and these are called antiviral strand proteins. And what they do is they have a nuclease that cleaves viral DNA, and as a result can defend E. coli, for example, against viral viruses of bacterial origin. Now, just to give this some perspective, because I know this is a very broad audience, that these genes also have been genetically linked to diseases. And I know Paul Liu has done a lot of work looking at genetic linkage to diseases. And the type 2, C2TA, I already mentioned, is linked to type 2 bare lymphocyte syndrome. NOT2 is one of the best genetic defects in Crohn's disease. And this protein, which is well known at NIH, and I'll go into this a bit more, called NLRP3 or cryopyrin, has been linked to multiple autoinflammatory syndrome. And autoinflammatory syndrome is really coined here at NIH by Dan Kastner's group. So what is this family of diseases? And these are all gain-of-function mutations, and that NLRP3 is important for inflammation, but when you have a gain-of-function mutation, this leads to severe rheumatological diseases. And this is related to increased IL-1 synthesis, which I will get into a little bit. And the treatment is IL-1 receptor antagonists or antibodies against IL-1. And this initially was the discovery of a young faculty at the time, not to say that he's old, but it's been quite a few years now, is Hal Hoffman at UCSD. And Dan and his colleague did a ton of work right here with Ilona and Rafaela, did a lot of work on this disease family, including much more severe versions of this disease called NOMID or SINCA. And treatment, like I said, is antibodies or anti-receptor antagonists, IL-1 receptor antagonists that would target IL-1. And patients really get better within days, which is remarkable. While before they have rashes in the less severe cases to really CNS problems as well as severe deformation, in this case of the knees. So there are many mutations that have been found in this disease, and all can be treated by these antibodies. So that is really a triumph of translating the bench to bedside, and much of this work is really done at NIH. So it turns out these NR proteins, what they do is they form this multimer called the inflammasome, and it's a word coined by the late Georg Schaap. And this just shows that different NR proteins are the receptor for different ligands indicated here. And what they do is they activate. In some cases, we still don't know how this is done, but they activate the inflammasome receptor, and then it associates with an adapter protein called ASE, and then this recruits pro-caspase-1, an enzyme, to cause active caspase-1, which then comes down here and cleaves two important cytokines, pro-IL-1, IL-18, into their mature form. So this is the inflammasome protein. In the last few years, it's found that caspase-1 has another major target, which is gasdermin D, which then forms pores in the membrane to cause cell death. So one important concept in the inflammasome is it requires two different steps. The first step is called priming, and priming is mostly signal transduction leading to the transcription activation of inflammasome components. For example, TLR can prime this, TNF can prime this, IL-1 itself can prime this, leading to activation of NF-kappa B, and then leading to transcription of many of the inflammasome components, including IL-1, IL-18, caspases, NRP3, etc. And then these proteins are formed, and they also go through post-translational modification. That's still the priming phase, such as ubiquination, phosphorylation, etc. And then all these components are made, they're ready to be assembled, and at the second step is signal 2, which is activation, causing the assembly of these inflammasome different components into this large complex. And so this then causes IL-1 secretion and IL-8, and also I mentioned gasdermin D, which when it's cleaved by caspase-1 comes and forms a pore at the membrane. So now we have the mature form resulting in inflammation and in cell death. So these are two different components of the inflammasome. So we have, as the field has, too, looked at tons of functions of the inflammasome. And all of these that I mentioned have also been done at the same time, or maybe a little bit later by others in the field, or sometimes a little bit earlier than by others in the field. For example, we found inflammasome is very important for influenza. It's also important in autoimmune diseases that are T-cell mediated, such as in EAE, which is a model for multiple sclerosis. It's important for metabolic diseases, and our paper showed it's really activated by saturated fatty acid. This is one work done by Haitao Guo. He's now a professor at Ohio State. And then we have papers indicating its importance in neuroinflammation and also in inflammation that doesn't require T cells. In addition, we found that these are really important in colitis, colon cancer, etc. So the broad impact of inflammasome is in many diseases. For this reason, there are billions of dollars spent by pharma looking at inhibitors of this pathway, and many of them are looking promising. So when the COVID crisis started, we, like everybody else, had to close down our lab, and one of the postdocs in my lab really wanted to study COVID. And I said, we've never done this, and we have to work on BSL-3 facilities, et cetera. But Kat was really undaunted. So she took on wanting to look at the role of inflammasomes in COVID. And this is because there are good indications that in severe COVID patients, there are increased IL-1 and IL-6. And in multi-system inflammatory syndromes in children, one of the main outcomes is increases in IL-1. In long COVID, there's also sequelas of IL-1. Clinical trials of IL-1 and IL-6 blockers in severe COVID patients, however, are mixed. And there's a few good papers looking at IL-1 inhibitors in severe COVIDs where there are indicators of strong inflammatory components, such as not only in inflammatory cytokines, but also in coagulation problems. And that's where IL-1 seems to be useful. So she was interested in looking at this, and many of these studies were done after we started our projects. So what Kat was looking at is this system that was really interested in the first step of COVID, which is infection of the primary airway epithelial cells. And this is a human epithelial cell culture that's in the 3D fashion, done by our investigators at UNC, Scott Randall. So he invented this whole system to really look at other diseases in the CNS. And this is just a cheat sheet in case you want to look at signal 1 and signal 2. So there are different things that are activated inflammasome in signal 1 and signal 2. And what we have found in this whole system is airway epithelial cells can make IL-1 when they're responded and induced appropriately by signal 1 and signal 2. So in this case, PAM3-CIS is signal 1 and Valpor is signal 2. And she was able to see decent IL-1 secretion in these airway cells. So this was the first indication that this cell type can secrete IL-1. So we wondered if COVID-19, SARS-CoV-2 can participate in this process. So we were able to look at SARS-CoV-2 infection of epithelial cells, and they went up in time. Remember, this is log phase of PFU, and we were able to see that SARS-CoV-2 infection of these airway cultures that are 3D was able to replicate and increase in these cell cultures. However, when we look at IL-1, we saw that SARS-CoV-2 couldn't induce any IL-1 in these epithelial cells. And these are the ones that are expressing all the receptors for SARS-CoV-2. They're nicely infected, as indicated here, but they produce no IL-1, and this is simply a positive control. So then we moved on and wanted to see if what is the effect of SARS-CoV-2 on human PBMC. At this point, there were quite a few papers looking at induction of the inflammasome in PBMC by SARS-CoV-2, but most of these require other signals. For example, some require priming signals, some require antibodies, but nobody was looking at just the virus itself, and we just wanted to look at just the virus itself. What does the virus do to PBMC? And so what we noticed that the virus do get in, but they don't seem to be replicating actively, and that the virus actually very quickly goes down in PFUs. And if we look at expression, we saw the same. This is expression of the N protein of SARS-CoV-2, and it also went down. So the virus is not actively replicating in these PBMCs. And what we notice is when we look at IL-1, if we're looking at the red bar, which is SARS-CoV-2 by itself, there's also no IL-1 production. So by itself, it doesn't do anything. This is the positive control, this bar. So SARS-CoV-2 by itself, when it's infecting PBMCs, it doesn't induce the inflammasome. When it's infecting epithelial cells, it doesn't infect the inflammasome. So when can we get this virus by itself to induce the inflammasome? So what we look for is what does SARS-CoV-2 do to PBMC by itself? And so what we notice is when we just look at SARS-CoV-2 by itself, indicated here, it induced relative expression of all of the inflammasome mRNA, and this just shows you some of them. So IL-1, NLRP3, AIM2, which is another inflammasome receptor, all were induced by SARS-CoV-2 nicely. However, when we went to look at its ability to induce the inflammasome, which is you have to look at IL-1. So this is just step 1. IL-1, the protein, is step 2. So that's the activation step. What we found is if we just have SARS-CoV-2 right here in this blue outlined area, there's no IL-1. What is required to cause SARS-CoV-2 to induce IL-1? So here's SARS-CoV-2 by itself, and then we added signal 2 nigericin or signal 2 ATP. And now finally, we're getting IL-1 produced. And this is very respectable IL-1, about 1,000 to 2,000 picograms per mil. So it turns out SARS-CoV-2 and PBMCs can only be signal 1. It can't be signal 2. So this provided us with some mechanism of how this is done, that SARS-CoV-2 is just signal 1 in PBMCs. So what is the pathway? What is the pattern recognition molecule that causes SARS-CoV-2 to be signal 1? So we looked at different molecules from SARS-CoV-2, and then we detected this one, which is the envelope protein, the E protein, and this turns out to be a signal to induce all the IL-1's transcription, and this is shown here. So here is using E protein, and this is a positive control. We're just looking at the first step of inflammasome induction, which is IL-1 message mRNA production. And E protein was able to induce our mRNA production. So we would assume we need a signal 2, and would that be sufficient to cause IL-1 protein production? And indeed, that is what we saw. So we use E protein, and we use S protein as a negative control. We include a signal 2, which is ATP, and we were able to see that this now induced significant IL-1. So in PBMCs, E protein is the one that acts as signal 1 in inflammasome activation. So we were able to identify the specific molecule involved. So then we wanted to know, we would think in an infected lung, you have the lung cells, and epithelial cells are one of the components. And the second component would be the immune cells, and many of these would be PBMCs. So what we did is, this experiment was very hard to do with the human airway epithelial cells. So we used varicocele, which is a very good source of infection by SARS-CoV-2. So we used varicoceles and PBMCs, and we infected these with SARS-CoV-2, and nothing happens with IL-1, as I have indicated. So if you just have these cells separately, they can't produce IL-1, even though SARS-CoV-2 in PBMC can act as signal 1. When we culture these two together, however, all of a sudden we are now seeing very respectable signal 1. IL-1. So we were able to put the two together, suggesting that some interaction has to happen between PBMC and the epithelial cells to get signal 1. We were able to show that this is caspase-1 dependent because we use the caspase-1 inhibitor, and now we get nothing. This is not so much dependent on NLRP3 because we added an NLRP3 inhibitor, and it was only reduced by about 40%. So there's something aside from NLRP3, and we're working on that right now. We have some indication, but it's not quite ready for prime time. So it's really two different cell cultures have to come together to get IL-1. So we wanted to know, what is it in SARS-CoV-2 infected airway epithelial cells from human that's acting as signal 2? And what we found is, we just looked at the culture supernatants of these infected airway epithelial cells. We were able to find that SARS-CoV-2 releases germline DNA, it releases mitochondrial DNA, and in fact we can PCR out specific mitochondrial DNA from the supernatants. So these cells are dying and releasing DNA from the cells. And we know that DNA can be a signal 2. So what we did is, we again took viral cells as a representative of airway cells and added PBMC, and the two together release IL-1. That's because viral cells provide signal 2, PBMC provides signal 1. We wanted to know if this signal 2 is DNA, so we treated this co-culture with DNAs, and now the signal went way down. So this suggests that these viral cells can release DNA, and when you get rid of these DNA, the signal 2 is greatly reduced, and now we have reduced IL-1 expression. We then wanted to know if this really happens in patients, and we did all sorts of computational biology that I won't be showing you. We saw increase of IL-1 in severe COVID patients. We see components of IL-1 increase. We actually saw certain NLR proteins increase. So we wanted to know in patients that are controlled, that is, are healthy controls but have a lung disease versus COVID-19 infected patient in their lung, do we see differences in the amount of inflammasome? And we indeed saw that. And by doing that, what we did is look for what we call ASC speck formation. So ASC speck is formed when all the inflammasome components come together. They form a visible speck, as shown here, and we can count in patients that have COVID how many ASC specks do they have. And we were able to show that COVID causes a great increase in ASC specs. So just to summarize what I've told you, what we think is going on is SARS-CoV-2 infect epithelial cells. These epithelial cells then come down here to the lumens and then infect in the bronchi myeloid bystander cells. And these cells, when they're infected, they can produce IL-1 message, but this is only signal 1. And it requires signal 2, which is these cells, when they're dying, is one way. They release double-strand DNA, which then activate the inflammasome as signal 2. Then you get IL-1 release. Then these are released. And I didn't get into the sequela of once you get IL-1 release. We showed IL-1 can cause IL-6, and you get a circular amplification of the inflammatory loop. So I should say at this point that we also have a paper on the sequela of this, looking at cell death that follows this. But because I'm following just innate immunity, I will not be presenting that. But that's a really interesting finding, where we found the airway epithelial cells that are infected, they go through necroptosis. And it's when they die, the bystander cells go through apoptosis, and different kinds of cell death occurring in different cell populations. And that paper is now in Science Immunology. So I've talked to you about the role of innate immune receptors in infectious disease, and one that has been really impactful on all of us. So next I'd like to really move into innate immune receptors on microbiota, and I presented this actually last November here at NIH, so I know this may be a repeat. So I will mention primarily many of the NLRs as activating innate immune receptors. But it turns out there's quite a few of these that, for whatever reason, when they're present, innate immune signaling are reduced or attenuated. And this includes many of the ones that we don't work on. All 4 of these, we don't work on them. But other people have found that they interfere with, for example, in TBK signals, IKK signals, et cetera. In fact, in one of them, it was found that these are important when the sperm DNA are injected into eggs, and that's a process where you have a bolus of DNA going into a cell as foreign DNA, and you don't want to have an immune response or interferon response against those DNA. And NRP4 was found to inhibit TBK activation by foreign DNA, which is sperm DNA. So it becomes important not only in the immune system, but also in the reproductive system. So we have for a long time been studying NRP12. I have to find it. It's down here, this protein, which has an enigmatic role because it also has been found to activate inflammasomes under some circumstance. But in our hands, when we look, and we've looked for over a decade in some of the diseases, whenever NRP12 is there, it inhibits many of the processes that cause signal transduction, leading to pro-inflammatory gene expression. So I'll just focus on this NRP12 protein, which we have found to be an inhibitor of the NF-kappa B activation pathway and the non-canonical pathway. So wild type, we see nothing with these signals of PAM3-CIS and CD40 ligand, and you look for signal transduction because P100 is then processed to P52, and we don't see that. And here, when we don't have NRP12, this is activated spontaneously. And we moved this to many different model systems and really couldn't see much of the effect until we went to DSS colitis. And this is simply a colitis model induced by this detergent-like chemical DSS. And so what we found, wild type didn't do so poorly here, but the knockout did very, very poorly, indicated here. And if we look at subsequent colon cancer formation, we were able to show that NLRP12 knockout did very poorly, had lots of colitis-associated colon polyps. So this was found by our lab as well as by the Kaneganti lab within a month or two of each other. So years later, we repeated this model, and we were able to see again NLRP12 knockout had increased weight loss. So NLRP12, when it's there, protected mice from severe colitis, and we were able to see that survival was greatly lost in the absence of NLRP12. However, this was not as dramatic as this case, where we saw such dramatic differences. And the only difference between these two was our mice was moved to a very clean facility, and because our vivarium wanted to save money, we later discovered they almost gave the mice a vegetarian diet. So all of a sudden, we're getting much smaller differences between these two. So we thought this might be due to the microbiome, and we were so lucky in that Belfer Sarter, who started the first notobiotic facility that's NIH-funded in the U.S., is at our institute. And we found when we put these mice, re-derived them in germ-free facility, there's no longer any difference between these two. So this started our work in the microbiome, and Leo, who did the work, was so careful. He did co-housing, littermates, and fecal transplants. And unless a difference is found in all three, he discarded the data. So this is what we found is that when we profile wild type and NLRP12 fecal material, we were able to see that the knockout lost diversity, as shown here. And by coordination plot, they are quite different. The fecal material 16S look very different between wild type and NLRP12 knockout mice. And then we looked at only changes that are consistent between two different vivaria. This is the clean one, this is the dirty one, this is the clean one, and these are litter mates. And he was so careful. Many people do litter mates, they're really cousin mates, but he actually used litter mates and spent a lot of money on these because you have to really do sister brother pairings. And so by looking at all these, what we found is that NLRP12 knockout mice have reduced expression of these two orders and down to one family, Lactobacillus. And at the time, we were really novice to this. We didn't really know much about these different families, and it turns out there was a paper from the Broad Institute showing that in pediatric patients, there's a reduction in exactly these two orders and in Lactobacillus. So we were comforted by this. And so what Leo did is gotten 23 strains of Lactobacillus from Vincent Young at the University of Michigan. He was able to use NLRP12 knockout. They lost a lot of weight in a DSS model. He reconstituted these with Lactobacillus and was able to restore the weight loss, much of it. And the same here, this is NLRP12 knockout with just BHI as just bacterial media, very severe disease index indicated here. When he added back Lactobacillus, these mice got a lot better. So we thought this was really remarkable that you can put a bacteria back into these mice that are severely debilitated by DSS and restore them. We also did this in obesity and type 2 diabetes, which I will not show here, and they were also published, that Lactobacillus was able to prevent obesity and type 2 diabetes in NLRP12 knockouts. So we got more familiar to this, and then we started wondering if these different bacteria have other applications. And this is how we started this project. It's mostly because we were funded by NIH to look at ways to reduce radiation injury during a terrorist attack. So there's a big program. We've been funded for over a decade to look at this, and we have other projects finding that a TLR2/6 agonist is a wonderful mitigator of radiation damage, and it can have its effect even a day after the damage has occurred. But what Hao Guo in the lab wanted to know is what is the link between microbiome and radiation injury. So she was really looking out for this. And we all know, of course, radiation injury can happen during cancer treatment, but also during accidents such as a Chernobyl accident. And upon radiation damage at lower dose, you can see hemopoietic damage. At medium dose, you can see a GI damage. At high dose, you can see GI damage, but also cerebral vascular damage, which at this point, there's really almost not much that one can do. So we were mostly interested in this phase of GI damage and gastrointestinal damage. So what Hao did, she was on the paper, a co-first author on the paper, looking at TLR2 and 6 agonists preventing radiation damage. And what she noticed that mice that survived, these are control mice that didn't receive anything except for 9.2 grays of radiation. The control mice, actually there's 15% that survived. And this is what many of us who do bone marrow transplant will see, that there's a percentage of mice that will survive so-called lethal radiation. So she was interested in this group, and she was wondering whether there's any difference in their microbiome. So she took these mice versus control mice that were just aged normally. So these are the mice that were aged normally, and compared the microbiome of these two and found a profound difference in their two composition. And she repeated this again, did a second experiment, only because we have these mice in hand, and found the same thing. And when she profiled the 16S RNA, DNA, and there was a major difference between the two group of what we call elite survivors and these just controlled old mice. So she did several experiments trying to figure out if it's the microbiome, and this is the cleanest we have seen, which is she took the controlled old mice versus the elite survivors. They're all over 2 years of age by this point, took their fecal material and transferred them into germ-free mice. We also did this with just normal mice kept in SPF facility, but these are germ-free mice, and she found a very big difference between the two. That is, those are from elite survivors, receive fecal material from elite survivors. They did remarkably well after radiation. So these were all radiated, and then we followed their survival. And these are the ones that received fecal material from just old mice. And there's a big difference, and there's also a big difference in their disease index. So the ones that received fecal material from elite survivors did really, really well. And from those two recipient groups, she again profiled the microbiome. They were quite different as well. So these are second generation. They're the germ-free mice that received fecal material from elite survivors versus controlled old mice. And what she found was a number of bacteria were increased in the elite survivors, indicated here, including Lactobacillus. So we were agnostic at this point, and we just put a bunch of these bacteria into mice and pre-treated these mice by oral gavage and then radiated them. So these are the different mice that we treated, and previous studies has shown that lactobacillus can be helpful. So that's the green one. And we were able to show if you treat mice before radiation, lactobacillus was indeed helpful. But what was surprising was Lactobacillus was really, really good. So this prevented 50% of the mice from dying. And we were able to show that restoration of the hemopoietic system by Lactobacillus indicated here, from these vacuole hemopoietic system to more regenerative system, and also the GI from this leakage indicated here at the point of the arrow to mended leakage here. So there's very little leakage when these mice were treated with Lactobacillus in the GI tract. So there are therapies for radiation syndrome in the hemopoietic system, but there's no therapy really for GI syndrome. And we're actually actively now starting to look at some of these in patients. So in the Lactobacillus, we know that this group of bacteria can make lots of short-chain fatty acid. So out of the 23 strains, we were able to identify 3 strains that made very low amounts of short-chain fatty acids and ones that made very high amounts of short-chain fatty acid. And we grouped these three together and these three together, and we fed them to mice, and then we treated them with total body radiation. And the ones that received these were much better protected, as indicated here. So these are the high short-chain fatty acid, suggesting to us it may be short-chain fatty acid that's protecting these mice. So then we also wanted to know if this has any clinical relevance. I hope I have the slide. Yeah. By obtaining patient samples at Duke and at Memorial Sloan Kettering and also at Cornell, patients who have received radiation, and looking at their fecal material for the amounts of OTUs of Lactobacillus, Enterococcasia, Lactobacillus, and the combination of these three. And we look for patients with diarrhea that lasted less than 10 days, which is less severe, versus those that have diarrhea lasting more than 10 days. And those with less severe symptoms actually also have increased amounts of lactobacillus. So this, of course, is a correlation. We don't really say this is causative, but it supports the hypothesis that lactobacillus may be beneficial in this group. And so next we wanted to see if short-chain fatty acid itself has any benefits, and we found this one, which is propionate, has really strong beneficial effects for radiation. Here we gave short-chain fatty acid containing water, and then we radiated these mice, and we showed that propionate pretreatment was able to really reduce, increase survival rate and reduce the disease index, clinical scores. And it had an effect on both the bone marrow, causing bone marrow restoration, as he indicated here. And I don't think I have the slide, but it also caused GI restoration. So in addition to short-chain fatty acid, which is well known in the field and has been shown by others to be beneficial in radiation, we wanted to know if we can do an unbiased approach to look at other metabolites that can benefit radiation damage. And we found tryptophan was highest on the score. That's different between survivors, between elite survivors and just old mice. And this was done by looking at the metabolites done by Kun Lu at UNC. And so we used two different tryptophan metabolites. One is from the bacteria and one is from the host. And looking at these two, we were able to show that treating mice, this is 1 day before radiation exposure, these two were able to reduce disease index and increase survival rates. And this lasted for over 300 days. So this is about here. So in conclusion for this part, we started from innate immune receptors, found a group of bacteria that we've been looking at, and found that these bacteria, including Lactobacillus, can also reduce radiation damage. And specifically, we were able to find metabolites downstream of these bacteria that can prevent radiation damage and restoring both hematopoiesis and intestinal recovery. So one of the young clinical faculty at UNC has used this data now. We started a clinical trial using short-chain fatty acids, propionate, and different combinations to look at its effect to reduce radiation damage. I want to conclude by saying, and not showing you very much data, that we always think about receptors as being cell type specific or adaptive immune specific or innate immune specific. And most people who are looking at innate immune receptors really have not looked at their functions in T cells and B cells. There's a small number of papers published on this, but we noticed when we're profiling, and many of the innate immune receptors are highly expressed in T cells and B cells. So these are some of the studies that we have done showing the importance of innate immune receptors in T cells and B cells. For example, this one actually prevents overzealous CD4 T cell responses in both virus infections. In this case, it's LCMV infection and autoimmunity. So what it does is eventually shuts down the pro-inflammatory response. We know this protein actually promotes oxidative phosphorylation and increases HIV replication, and correlated with large patient samples where HIV is increased correlated with increased oxidative phosphorylation signal. And finally, in this case, we found AIM2 has a profound effect on preserving T regulatory cell stability. So different NLR proteins in AIM2 have roles in preserving T cells and B cell functions. In addition to this, this is a well-known pathway, which is CGAS and STING. CGAS detects DNA, indicated here, produces cyclic AMP and GMP, and this cyclic CGAMP activates STING to cause interferon and other pro-cytokine approach induction. And this is found to have many different roles, including promising effects in many cancer preclinical models of cancer, because you would get lots of interferon production, lots of CD8 activation, et cetera. And this would go on both in the myeloid cells as well as the cancer cells themselves. However, when this is used in monotherapy of STING in cancer for clinical trial, the results have been very disappointing. And the other thing is, in most mouse model, STING has to be injected inside the tumor. So this would be very difficult to execute. And we have a paper showing that what turns out to be the case is that STING can activate B cells, but a population that you probably don't want in cancer, these B regulatory cells. These B regulatory cells are induced by STING activation to produce an immune suppressive cytokine called IL-35, and IL-35 then reduces NK proliferation. And so STING can have this other negative effect on the immune system to block NK cell cytotoxicity. And this was published a year and a half ago. So we wanted to come up with a therapy, and one of the therapy that Wei-Hei Liu and his postdoc, they were kind enough to recruit us as the immunologist. So this paper was just published in Nature Nano. And what they have done is taken the natural configuration of CGAS complex with CGAMP, and it is a tetramer. So what Wei-Hei has done is take full-length STING and conjugate it to an oligomer, an artificial tetramerization domain. So this forms a tetramer, and you have an activated STING that would go to cancer cells. And what we have found is we've looked at both human and mouse cancer, and this was able to go into all sorts of cancer cells. And so what we do know is that cancer, even though STING and cancer can help in the STING therapy, it turns out a majority of cancers actually have lost their STING, whether it's inhibition of STING pathway itself or downstream signals such as the IRF3 signal. So in most cancers, there's no activated STING. So we wanted to know if we can use this uni-STING to activate STING in cancer. And what's unique about the uni-STING, this tetramerized STING that was delivered in lipid nanoparticle as an mRNA, is that it induced the interferon pathway as indicated here, but did not induce much of the NF-kappa B pathway. We still don't know why, but NF-kappa B can be both pro-inflammatory and it can also be pro-cancer. So way back 20 years ago, many labs have shown NF-kappa B is actually pro-tumor, including the lab of my neighbor, Al Baldwin. So that was shown years ago. So in the uni-STING, it also only induced interferon, and we wanted to know if this uni-STING can be useful in different tumor systems that do not have STING activation. So this is one of the cell lines. This is a triple-negative breast cancer, and this is a metastatic liver cancer. In both cases, if we just deliver STING agonist, the 2,3-CGAMP, it didn't do very well, and this is delivered systemically. And if we deliver uni-STING systemically in lipid nanoparticle, it did really well. And we have followed it up to about 60 days in these tumor systems. So just to conclude, we think many of these innate immune receptors, such as STING, can activate regulatory pathways. But this may be one, the uni-STING, which is artificial STING, may bypass some of these regulatory pathways. It's a self-assembled tetramerized STING. It's delivered as a lipid nanoparticle. These particles are not known to go into T cells or B cells in high amounts, and it was tested in multiple cancers, including many of these. It does not require endogenous active STING in the tumor and activates the IRF3 pathway without activating the tumor-promoting NF-κB pathway. So in conclusion, I just want to show you that this is innate immune receptors can have many different effects, including its effects on infections, inflammation, autoimmune diseases, metabolic diseases, cancer, DAMPs and PAMPs, as vaccines, affecting microbiome, and also in T cells and B cells, which were found to have many of these innate immune receptors. So I just want to really thank the people who are involved. Our early work on NRP12 was done by these former members. 2 of these are now faculty at different institutions, and 2 are in pharma. And these are the many people who studied innate immunity in T cells and B cells. Again, 3 of these people are now faculty in Japan as well as in the Netherlands. And Kat Barnett, who's going to the University of Michigan as a faculty, did all the SARS-CoV-2 work, and let's see, how did all the the microbiome work together with June Brickey, and these are some of our collaborators. Yulia helped us a lot with the B-cell work that I mentioned, and Balfour, I mentioned him. Vince Young, I mentioned his name, at Duke, Solon Kettering, and also at Cornell. And then finally, in another acknowledgement, these are the 2 people who, despite COVID, went to Duke. For all sorts of reasons, we couldn't use our BSL facility at UNC. So we had to go to Duke. They drove 25 miles to do all these experiments, and they got trained. And this is Kat Barnett and Kai-Shen Liang. And these are the many investigators who helped us in Marsico, the lung culture. I mentioned Scott, Duke, WashU, University of Michigan, and NIH especially. And Yu Ying, who helped us with a lot of computational biology. Thank you very much for your invitation. It's a great honor.
There's microphones in the steps corridor.
I think I was told that online there may be questions.
Yeah, we got that covered. Yeah.
I had a question about the radio protection story.
Yeah.
Is there any benefit if you combine the short-chain fatty acid with the TLR2/6 agonist?
Yeah, we're actually trying to do that because the short-chain fatty acid, it has to work beforehand. So it's great for cancer patients, right, because you know they're going to come in for treatment. We actually enrolled at least 11. We prepared the samples for the clinical lab. We have quite a few who already came in and gave samples. So we would know that soon. Yes, Jake. Oh, sorry.
Oh.
Jake.
Hi, over here. Among the type 1 interferons, there's some different functions between alpha and beta.
Yes.
And I understand that interferon alpha that's made primarily by PDCs as opposed to interferon beta that's made by many cells
right.
when you induce with STING and so forth, may be more effective, at least in humans, than inducing CD8 T cells. And may be more effective in cancer. And so the question is, does this unisting activate PDCs to make interferon alpha? Do you have any information?
Yeah, that's a great point. We did not look. We did look at myeloid cells. Didn't really focus on the PDCs. We focused on interferon beta, not so much alpha. Mm-hmm. But because we were mostly interested in CD8s, and I didn't show you, but this unisting is entirely dependent on T cells. So we get rid of CD8s, it doesn't work. So most likely it's beta, but we didn't look at PDCs.
Yeah, apparently there's a difference between mice and humans in that regard, that alpha and beta have more differences in human than they do in mice.
Yeah, so we didn't do this obviously in humans, but we did look at human cancers to see if it has the same effect. And for the parameters we were measuring, such as interferon beta and all the signals, it did exactly the same. So we used 2 different human cancers and I think 3 or 4 different mouse cancers. And the effect on interferon was similar. And we did a lot of Western blots, which I took out all the Westerns. I thought people would be going nuts. Thank you.
Thank you. Mhm.
Yes.
So Jenny, thank you for such a fabulous talk.
Thank
My
you.
initial impression listening to you talk was, wow.
Thank you so
really
much.
covers so
That's
many
so kind.
areas and clinical and basics. So I was just intrigued by your observation that many of the inflammasome components, such as NLR3, that actually are blocking the interferon induction response. I wonder whether, two short questions, I wonder whether it has any effect on the downstream interferon effect. You mean after step 1, 2 activation? Does that block that as well, or is it more at the induction
Yes.
of the interferon?
So we do see stat changes, but that's we imagine it's from interferon.
Upstream.
Yeah.
Okay.
And we have a NLR called NLRX1, and we're reviving interest. You always have to cajole people in the lab to work on projects, but we have one that actually blocked MAVs and the MAVs rig-I function, and there's a number of labs that have shown the same. But again, that's upstream. So what we're looking at is mostly upstream. We have not seen downstream inhibition. We did see TBK, or we didn't. Other people have TBK inhibition. So
So it's possible.
yeah, it's possible.
The other question is, since induction interferons actually play a major role in vaccine
Yeah.
effects, so I wonder whether the inflammasome may be involved in some of the vaccine response, and whether there's something would be worthwhile to think about, whether you block the inflammasome maybe will enhance vaccine efficacies. Or,
Yeah.
I
So
mean,
one of the best one is, don't think anybody has been thinking that way, but
see.
one of the best example of inflammasome blocking interferon is AIM2. So that's been observed by so many people. You activate AIM2, interferon goes down. But I can't think of a single study that's using that to take advantage of it for vaccine purposes.
I see.
Probably because all these other cytokines, like IL-1 is probably necessary for vaccine response as well.
Yeah, sure. Okay, thank you.
Thank you.
I think that will be our last question. Thank you very much for attending, and we'll have a reception just outside as your reward for showing up in person. Another round of applause.
Thank you.